So our next speaker is Dianne Gleeson, and Dianne is from the University of Canberra, and she's speaking on environmental DNA and wildlife surveillance from promise to delivery.
Aha. Yes. Well, thank you. Thanks, Neil, and great to be here, and been really great catching up with everyone, and thanks to the speakers prior to me, because you've kind of outlined a lot of things about eDNA, which is fantastic. So what I'm going to talk about is with my colleagues Stephen Saar at the Institute for Applied Ecology, University of Canberra, and my postdoc Elise Ferlin. I also need to acknowledge the support of the Invasive Animal CRC, which has been really instrumental in developing the technologies, or supporting us to develop these and delivering them, and the new iteration that we're all going to be basking under, the Centre for Invasive Species Solutions. So our CEO is in the audience, Andreas, thank you. I also need to acknowledge the team before I carry on. So I've got, so there's, I'm just making sure I've got the thing. Oh yeah. So the lovely Elise Ferlin, she's been the amazing postdoc that's really sort of driven this work. My PhD students, Jonas, Rada, just to show you that in Australia we do get out into extreme environments, and we can, you know, that's the eDNA on the edge. And of course the wonderful Professor Richard Duncan, who's hopefully floating around here in the audience, and I encourage you to go to his talk tomorrow. And Richard is the driving force around the real guts of what we need to do in terms of parameterising, having quantitative measures, and embedding this in some kind of modelling framework. So I appreciate that, and all our funding agencies and CSIRO, who are now welcoming us into their bosom of the Future Science platform. So thanks, you guys. So yes, it all looks really simple, and that's a really nice, lovely, simple slide. Thanks, Andreas, for that. So just illustrating the 4 species that we've been kind of focusing on for our eDNA work. So obviously eDNA comes from a whole range of different substrates, can be soil, fecal matter, and we've focused with this particular work on water, but other areas in our lab have focused on other things. So I'm going to talk about the watery side of it. So it all looks fabulous and easy, and sort of Gavin kind of pointed out, you kind of grab your sample, you extract your DNA, and away you go. But that's not necessarily the case. So we've spent quite a bit of time trying to deal with some of the issues and variables in regard to environmental DNA. And I think this is something that really has to be nailed in all environmental DNA studies, because I have a real problem if people are not addressing this in their studies and making it really clear what how they've dealt with this, and can really get to the sensitivity levels that we need to know. So here's a simple situation where we've got our target species, it's chucking out DNA. The whole rates of DNA production can change, can depend on the season, can depend on what the animal's doing. Is it stressed? Is it reproducing? Is going to contribute more DNA to the substrate at that time. Then there's also the rates of eDNA degradation. Somebody asked about how long does it last in the environment. Well, we've done things where we've shoved fish into cages and dropped them into water where they're absent from that system, and we've been able to say, okay, in a flowing system, it's gone after about 24 hours. So and then the subsequent removal of the animal. Then there's the things about concentration and dispersion of DNA, because DNA is clumpy in the environment. So it might be that you kind of take 1 or 2 samples and you don't get the DNA, but it does exist there. It's just that you haven't sampled enough. You haven't got enough water. You haven't got enough of your substance to be able to say what is your probability of detection. So this is really absolutely critical to any DNA study, is understanding that. So all of this, and the type of sampling, like the numbers of samples you need to take, and then you extract your DNA, and then how many replicates, like in terms of single species detection, which is the work that we've been doing on invasive species, is, well, how many aliquots then? Because that's clumpy there too. And so all of that, so sensitivity depends on all of these things. And so we need to have those frameworks before we can go out and really go and use this as a surveillance detection tool. And just a little plug for the work that Elise and Richard and myself have done looking at this in terms of developing a framework so that we can get to the sensitivities of eDNA. And just a little idea, just to show you, with our 3 target species, these are all 3 invasive species in Australia. So we've got the carp, European carp, oriental weatherloach and redfin perch, and you can see here, so we've got sampling period in autumn, a sampling period in spring, and there's real differences in terms of here's our sensitivity up the side, and if we're wanting to obtain a 95% detection probability, here's the number of water samples that we would need to take, so this is the modelling side of it that's really important, to be able to get to that 95% detection probability. And you can see that, so things like carp, you would need to take about 9 samples. For oriental weatherloach, you need to take about 15 samples, and for redfin perch, you're going to need to take a lot more, up to 20 samples, about 18, 19 samples for redfin perch of your half-litre water samples. Come to springtime, whole different story. We only need to take about 4 samples to get carp, about 3 samples to get weatherloach, and about 6 samples to get redfin perch. And you can see, okay, so if you're going to design an experiment to go out and do surveillance, that obviously spring's your best time to go and do that. It's much more cost effective, and also that it relates to the biology of these species. Things are warming up. Species are becoming a lot more active, a lot more reproduction going on in the environment, a lot more eDNA going out into the environment. So what we've done so far with our work, and this is through the invasive animal CRC, we've been able to demonstrate the effectiveness and the efficiency of eDNA in the field. We've done things like, this was a project where we were looking at the incursion front of an invasive species, which is redfin perch, into a creek system. This is Blackney Creek. Now, electrofishing, traditional sort of electrofishing surveillance, which is what they would use, they found the incursion front to be here at this arrow. The green dots indicate where we've found it in terms of the eDNA, and this is incurring into a habitat of this pygmy perch, which is a highly endangered species. This is only one of 2 locations that this species of freshwater fish exists, and this redfin perch is a predator of that species. So they were going to place a fish barrier to prevent further incursions of redfin perch here, but clearly that would have been behind the incursion front, and so now the fish barrier has been erected here. So that has determined the optimal location of that fish barrier to prevent redfin perch incursions. We've also highlighted through work that Rada, my PhD student, has done in terms of monitoring, the monitoring methods for invasive species. So a lot of the species that have been used, detecting surveillance methods used by sort of traditional means, we've shown that, eDNA is showing the presence in habitats where the traditional methods have not shown them, so have actually missed them. So it is way more sensitive from that perspective. And then we've developed some novel applications of eDNA for detecting things like reproductive activity. So we know that we've got nuclear DNA, which is in your nucleus, and then there's mitochondrial DNA, which is then in the cells of organisms. And so what we'd expect to see, that because generally we target mitochondrial DNA, we'd expect to see in reproductive times, when there's more nuclei being exuded into the environment, that we're going to see a spike and a difference between those 2 types of DNA, and it can tell us whether there's reproductive material that's present in the water. And this is showing you in different densities. You can see that spike difference at a sampling period when we knew that there was going to be a reproductive event. So this is great for a conservation species where you're trying to locate reproductive activity and a location of where that happens. This work, which we've done in the Murrumbidgee River recently, so this is looking now at whole community analysis and whole community diversity. So we started sampling up here in the higher altitude areas of the Murrumbidgee River. We would expect to have less species diversity, through to sites where we're expecting higher levels of species diversity. And so this is now taking whole samples and looking for all the species, so reconstructing species communities. So this is the inventory that we gained from our eDNA project. And we can see that we've got lower levels of diversity in terms of numbers of species across our sampling sites. This is the high altitude through to the low altitude. And you can see the big dominant one here is carp. So carp is just throughout the system, and it is definitely the most predominant sequence. But if we drill down into this area of our numbers of sequence reads, we can see here we've got a lot more of the species diversity and a lot more of the native species. And we've actually picked up species that have not been found through other methods and means as well. And that leads me on to carp. So carp has been a big issue in Australia, and The silver bullet, well, this may not be quite the silver bullet, but there is a technology, biocontrol technology that's been slated to be released. There's been a huge amount of funding put at it, which is the Carp herpes virus. So this has been trialled, tested really thoroughly by CSIRO, found to be incredibly species specific, and they're looking to release this into the environment in 2018. So yes, been tagged as Carpageddon by our minister, the Agriculture Minister, Barnaby Joyce. Now, Barnaby has gone mad in Parliament over this time, and he had quite the rant and about how many tons of carp. He kept yelling carp, and it was very funny, and so a lot of amusement. And there's been even more amusement, of course, by everybody about saying, well, this is what's going to happen. We're going to have carp. We're going to figure out what to do to avoid catching herpes, because they're releasing herpes virus into the environment, and this is all shocking and terrible. So anyway, it's caused a huge amount of interest. There's a lot of money being put at this biocontrol agent, and that's been kind of great for us in terms of researchers, because what we're really interested in, and somebody asked that question too, can you associate eDNA with biomass? And this is like a real major thing. As you saw what Barnaby was saying, there's going to be thousands of tonnes, hundreds of thousands of tonnes of carp washed up, and what are we going to do with them all, and how are we going to be able to predict where that's going to happen? So we've been involved in in trying to parameterise this, so this is where we're at the moment with our work, and we've been lucky to get involved in projects where they've been draining lakes, so they've been physically draining water bodies, they've been grabbing the dead carp, counting them, weighing them. Some real biggies came out of this. These are some of the ponds near around the ACT, and so they've been monitoring using traditional surveillance, so using electrofishing and sonication, and we've come in and done all our eDNA sampling with the idea that we can get to a biomass index that will be really, really useful for when this carp herpes virus gets released, not only the detection in the biomass, but also predicting what's going to be outcomes for carp. And just to show where we've kind of got to with some of this work, this was work down in Tasmania, where carp is not present in the environment there, other than 2 locations where it had been introduced. So it was introduced in Lake Crescent, but they've now managed to eradicate it. And so these are large water bodies. So you're talking sort of 37,000 million litres of water, and Lake Sorell, which is adjoining it, is a much, much bigger lake system. Now, they eradicated from Lake Crescent, but that was through traditional surveillance, and so they wanted to see whether the eDNA could back that up. Then they've estimated now through this has been a long period and lots of money being put at this carp control program. They're estimating that there's about 1,100 to 1,840 carp remaining. So we went out to see what we could do in terms of eDNA. So out of 100 water samples, we had 2 positive eDNA samples from that. And in terms of positive eDNA replicates, 600 replicates done of our qPCR, and we got 2. So we said, okay, so you had to take 100 times 600 mil samples per lake and take those replicates, and this is the kind of result that we got. So we're talking really low densities and we could get to. So from that, then Richard can come and weave his magic and come up with a estimation of how many samples would be required if carp, if let's say in this case 200 carp remain in Lake Sorell, how many litres of water will we need to sample? So we estimated through modelling it's going to be 7,144 600 mil eDNA samples are required. Now when we did that and we went back to the Inland Fisheries Service in Tasmania and told them that and how much that would cost, they went fine, because in fact the cost associated with traditional surveillance is so much more, and the time taken, that this sort of eDNA method didn't freak them out. So we were kind of pleased about that. So I just want to end on some work that Elise is now doing in Central Australia, where we're looking at water holes. And we're now doing a surveillance of all species that come and visit those water holes, regardless of whether they're a aquatic species or a terrestrial species. And this is really useful for looking at if we can use water holes as a way of doing wildlife surveillance for invasive species, things like, as you can see, some of the species we picked up, camel and wild horses. And so she's shown that this is really, really useful, but we've also got to be really careful about how we interpret the results and the kinds of workflow that's required to be sure that you're not getting somebody's tuna sandwich in the water, which is often the case with eDNA. You've got to be super, super duper careful. So here's my crazy ambitious final slide, which is what I believe is the future of eDNA sampling and processing, and we're kind of almost there with some of this. The miniaturization of sequencing through the Oxford Nanopore MinION, which is a tiny little device with a USB port plug. You just basically shove your sample in there, sequence straight to your computer, and that is ready. That is ready now, and so we're starting to trial that work thanks to the Centre for Invasive Species Solutions. That's where our next project's going to be focused on. Then you've got autonomous sampling devices. Here is, what did I just do? Help? Oh, good, thank you. We have the SAFA, which is CSIRO in Tasmania and Hobart. This is an automated sample filtration and archival device. It can do in situ sampling, in situ filtration, collects samples, 24 samples at a time, so it can collect them at every interval that you want to program it to do. It stores it in the filter, and then you can go on and do your extraction. This was deployed on last Tuesday, and so we were very excited by that. Then there's the MOBI, which is actually a real-time algal ID. It does, and this is in terms of oceanography biosensing instrument. It is fully automated. It does the whole in situ DNA analysis on waterborne microbes. And so this is another thing that's still just, you know, in the final throes of being developed there. So I can see that we're going to have automated samplers across sort of global arrays. Those global array samplers, and we can have this sort of in-cloud network reconstruction, so taking all that information, building the networks, we can then enables us to analyse, we will be able to analyse across highly replicated networks to detect changes in systems, and so looking at network interactions between species. So I think this is the crazy ambitious, but it's not that crazy or ambitious because it's kind of there. Thank you.
So, first of all, I guess it's really interesting study. I think it's really, really important as well that we can, thank you, when we are doing these eDNA analyses, that we have really strong, rigorous approaches so that when we do or don't find an eDNA signature, we actually know what that means.
Yes.
When we find something, sure,
yes.
we have an idea of what it means, but
yes.
it's really easy to get false negatives, and
Absolutely.
I think that these kind of approaches are really important. The question that I wanted to ask is, have you considered, you've got some really great eDNA data from your Carp, from your qPCR. You've also got the ability to do your DNA sequence analysis of the total communities as well.
Yep.
Have you thought of matching the two together so that you can correlate the impact of the biomass of the carp
yep.
on the composition of the native fish
Absolutely.
communities?
That's exactly what we're doing, yeah, and that's going to be the key to seeing how this rollout of KHV is going to then impact on whole community analysis. And yeah, but anyway, Neil can tell us about Loch Ness monsters, can't you? And,
I want some samples.
and you can have your sapper. And you
yeah, I want 10 of them.
You want 10. They're $100,000 each, apparently.
Can I get them
but
free?
if you're a friend of Cyro, and in my case, I'm a close friend, it's free.
Right. Well, on that note, thanks very much, mate.